The overall goal of this procedure is to assess the effects of amyloid beta toxicity on neuronal function and survival. This is accomplished by first constructing transgenic C. elegans worms expressing an amyloid beta peptide with a GFP reporter in ASE sensory neurons. Next, the behavioral consequence of expressing amyloid beta in ASE neurons is assessed by a chemotaxis assay. The third step is to obtain primary cultures of ASE neurons and then treat cultured neurons with pharmacological agents such as QVD-OPH to suppress the toxic effect of amyloid beta. Ultimately, results can show the effects of amyloid beta toxicity on ASE neurons' function and survival through behavioral and pharmacological assays.
This method can help answer key questions about neurodegeneration, such as elucidating specific cellular and molecular mechanisms underlying the neurotoxic effects of amyloid beta oligomers. Demonstrating the procedures will be Dr. Zhibing Duan, a postdoc from my laboratory.
For the transformation, 2% agarose injection pads are required. Place a drop of hot agarose dissolved in water onto a glass cover slip and quickly place a second cover slip on the drop and lightly tap it. When making the injection mix, be sure to centrifuge it and transfer the top 5 microliters, free of debris and other contaminants, to a fresh tube for injection. Load injection mix and empty plasmid into an injection pipette by capillary.